Hello, Facebook. I'm Evan Deshevsky, features editor with PCMag.com. Welcome to The Convo. This is the Facebook Live series where we talk to the boldest mind in science, technology, and geekdom. Okay, so today we're going to talk about conservation after the fact. Now, humans have done a lot of damage to other creatures that we share the Earth with. Sorry about that, guys. Now, why this damage has accelerated in recent centuries? Man-made extinctions are actually nothing that new. We've been making species extinct since prehistoric times. Now, but recent advantages in genetic technologies may allow us to undo some of this damage, including bringing back extinct species through cloning. How cool is that? Now, this all may sound reminiscent of a certain 1993 sci-fi blockbuster, but that's probably not going to happen for reasons that we'll get to in a bit. Now, to help us sort this all out, my guest today is Ben Novak, the chief scientist with Revive and Restore, a nonprofit project currently working on using genetic technologies to help endangered species, but also to bring back extinct species through cloning, de-extinction. Ben Novak, thank you so much for joining the convo.
I'm glad to be here.
Okay, now also the important part of this is we get the input from you through Facebook. Social Pete is looking at all the comment streams, so leave your comments there and we'll read them off later in the show. So, Ben, now humanity has been cloning animals since Dolly the sheep in, say, the mid-90s, but what technologies have come along in recent years that have made your project possible?
So what's going on in the 20 years since Dolly? Mm-hmm. The main advance that's happened was in 2012. Mm-hmm. Cloning, reproductive technologies, those have all been marching forward. People have been doing genetic engineering for over 30 years, but in 2012, the technology that changed the game was something called CRISPR-Cas9, mm-hmm. which now lets us not only add genes in or cut genes out, but lets us actually rewrite the code of DNA so that we're not messing around with it in unknown fashions. So now we can take the DNA, say, of an elephant and just rewrite it so that it actually matches the code of a mammoth. Mm-hmm.
Now, one of the projects you are working on is Mammoth, which we'll talk to, because I'm actually personally, for personal reasons, kind of really excited about that one. But before we get there, now, humanity has brought back extinct animals. Now, a Spanish team brought back something called the Pyrenean ibex, but it died shortly after birth. Now, these sort of problems actually have happened throughout the history of animal cloning, where the organism makes it to birth, but then dies shortly thereafter. What kind of why does that happen with cloning?
People believe that most of the problems associated with cloning occur because the genome that you're inserting into the egg cell mm-hmm. to give you your offspring, the egg cell has problems reprogramming it to the epigenetic state that it needs. And interestingly enough, cloning has come a long way to the point where for some species, such as mice or cows or horses, that it's so routine that they don't really expect those types of problems anymore. Mm-hmm. And they've been working on ways to advance that programming. I actually just read an article last month that came out this summer that Dolly the sheep, mm-hmm. even though she died young, mm-hmm. people all thought, oh, it was problems with cloning. Well, they cloned Dolly the sheep 4 more times, and her 4 clones of a clone, mm-hmm. are alive today and healthy. Wow. And the entire difference was the fact that Dolly was raised indoors, mm-hmm. and she developed problems from living indoors, and her clones were raised outdoors where she was supposed to be, and they're completely healthy.
Wow. Just for anyone who's joining us, my guest today is Ben Novak. He's working with an organization that wants to clone extinct species like a mammoth or like a passenger pigeon and a few others for ecological reasons. It's really cool stuff. So drop your questions, and Social Pete will read them off later in the show. So, okay, you have Revive and Restore has 4 main de-extinction projects that you guys are working on. The black-footed ferret, the health hen, the passenger pigeon, and the woolly mammoth. So what makes a good candidate species, and why won't we get dinosaurs like I and so many other people would really want you to be working on?
So the reason you're not going to see dinosaurs is mainly the concept of de-extinction is about actually working with the genetic code of an extinct species. It's not about creating simply a look-alike that has nothing in common with the extinct species. And dinosaurs, they just simply went extinct way too long ago for us to retrieve any DNA from their fossils. And actually, when I was in my early 20s, I worked on dinosaur fossils and you know, tried to figure that out. And it just, there's nothing there. DNA might last around 1 million years if it's frozen in permafrost, but otherwise it degrades really quickly. So we're restricted to being able to work with things within about the, 10,000 to say 100,000 year time frame for most of the places on the planet.
Now,
so that pretty much means mammoths and passenger pigeons.
So just to give a little pushback, because I know the audience is going to want me to do that. So even like in Jurassic Park, they found the blood in the fossilized amber. That's not going to work for you guys.
No, it's not going to work. People have tried many, many years to get that to work, and it just it fails all the time. So no dinosaurs, but there are teams working on projects to re-express dinosaur traits in living birds. Guys like Jack Horner, who actually was the advisor to Jurassic Park. So, maybe by the end of our lifetime, people will have recreated Chickensauruses and bred them to look more like velociraptors or something like that. But that's definitely not really de-extinction the way we're doing it.
So let's talk about the projects that you guys are working on now. Your specialty, the special team you're leading, is for the passenger pigeon. Now, give a little background on what that species was, because they went extinct just in the last century, and why you're trying to bring it back, and also how you're trying to bring it back.
Yeah, the last passenger pigeon went extinct in 1914, and give me some clarity. Where are you geographically located right now?
We're in New York City.
New York City?
Yeah.
That was prime flying grounds for passenger pigeons about 150 years ago. New England, the eastern United States, that's where passenger pigeons were native. And they were a really beautiful, unique North American pigeon. Slate blue backs on the males, red breasts. And what they're most famously known for in history is they were the most abundant bird species on the planet, flocking in these huge, huge masses of hundreds of millions to billions of birds from one area to another. And they were a real ecological force of nature.
Yeah. So what happened?
So because they flocked so densely, that it was really easy to kill a lot of them at the same time. I think the record is some priest actually shot about 132 of them with one shot.
Wow.
And people just did that. They would throw them up. Joel Greenberg's book has a whole lot of different ways people killed them. And at the time, food markets actually made an industrial scale market out of harvesting them, and hired people to go out and harvest their food, and they became a cheap source of meat. And over the course of about 40 years of relentless harvest, amidst a period of deforestation, the birds were just completely killed off. And by the 1890s, there's just a few stragglers, and by 1902, the last one was shot in the wild. Wow. And it was just a few left in captivity that lingered on and then finally died in 1914.
So what's your plan to help bring them back?
Well, so the reason we want to bring them back, which is probably worth mentioning is, because in true Jurassic Park style, we want people to know that we're thinking about why we should do this rather than just if we can do this.
Right.
So we've been researching passenger pigeons, and they play this very important ecological role of causing forest regeneration, making a mosaic habitat that living species today actually need, and many living species in the East are in decline because this process is gone. And to get that process back, we figure, well, let's get the bird And that starts with analyzing DNA. We can get that from museum specimens. If anyone in New York goes down to the American Museum of Natural History, you can see a few passenger pigeons on display. And we just cut a little piece of tissue from the toe, dissolve that up, filter the DNA out, sequence it. Right now we have nearly the entire genome of several passenger pigeon individuals, over 990 million base pairs of code for each of them.
Wow.
And we compare that to the band-tailed pigeon, which is its closest living relative, figure out all the interesting bits that make passenger pigeons what they were versus a band-tailed pigeon. And then the next step is that we'll start using CRISPR-Cas9 to edit those interesting parts into the band-tailed pigeon. And over the course of a few generations of cell cultures, which happens in just a matter of weeks and months versus years in birds, we can make a genome that's more and more like the passenger pigeon. And then we can't actually use those cell cultures for cloning in birds because it's just a horribly difficult process in birds. But what we can do is take those cells, introduce them into kind of a surrogate parent bird. And when we breed a male and a female, they'll unite the cells that we gave them. And out of the egg hatches a newly engineered passenger pigeon.
Oh, very cool. Facebook, I see you have a question.
So we've had a couple comments from people who are concerned that bringing back species would cause some sort of havoc. Someone mentions it would disrupt nitrogen cycles, or just in general, like just the ecosystems of wherever they were brought back into.
So with any form of de-extinction project, the reintroduction of an extinct species goes through a process of analysis, risk assessment. And this is similar to what people have already done with other projects. People don't often think about this, but the concept of de-extinction has been going on for a long time. The wolves reintroduced to Yellowstone represent the reintroduction of an animal missing from an ecosystem for 100 years. People have introduced beaver into Scotland experimentally and gone for 400 years. And these were intentional things done because the ecosystem was seen to actually need the transformation that species bring. So will it disrupt nitrogen cycles? Oh yeah, and that's exactly what we want with passenger pigeons. So think this way, passenger pigeons are moving around, depositing a lot of guano in one area of forest to the next. They were an active part of moving nitrogen and phosphorus cycles. Extinct, that became disrupted. So by disrupting the current nature of this, we're actually enacting restoration.
Very cool. Facebook, you have another question.
Is cloning animals legal in the U.S.?
Everyone watching today has probably eaten meat from a cloned animal.
Is that
the
true?
The FDA does not regulate the selling and distribution of cloned meat because they did a lengthy multi-generation study of cloned animals in agriculture and found that they are 100% the same as non-cloned animals, and therefore they're fine to go to market. It's a pretty common practice nowadays.
Enjoy your burger, everybody. Okay, so now on your website for the Passenger Pigeon Project, you say that you want to actually get to breeding them in captivity in the early 2020s. Are you still on that time frame? And then I believe release them into the wild in the 2030s. Is that correct?
That's our aim. 2022 is our goal for hatching a first generation of new passenger pigeons. And in truth, that first generation of chicks will probably not be the generation that would be ready to go to the wild. It might take some testing and refining of discovering what genes work with the environment and which ones are not really working with the epigenetics of band-tailed pigeons. It's a really complicated process of really neat discovery for us. And it would be awesome if by 2032 we were having some soft releases and considering releasing birds for trial periods and research. But it might be a little longer than that. But with support from the public, we can definitely try to hit that target.
Facebook, you have a question.
So people haven't really let go of the dinosaur thing.
Sorry.
We have someone who's asking, could you create look-alike dinosaurs?
So yeah. Theoretically, you could take a chicken and make it develop a long tail. You could make it develop simple quill feathers, or even potentially make it have scales over its body. But in truth, most dinosaurs probably had feathers. At least all the meat-eating dinosaurs had feathers at one stage. And you could get teeth back, remove the beak. In fact, people have already started these retro development experiments. If you get in the paleontology literature, people have managed to get embryonic birds to grow dinosaur ankles and have a dinosaur-shaped snout rather than a pointy beak. And they've gotten them to grow teeth and keep their claws on their hands and have little long tails. I mean, technically, enough people have done enough work that right now someone could make a chicken look like a dinosaur, but there's some ethical and scientific boundaries in the paperwork to get to actually letting one grow up.
Well, let's actually talk about another cool project is the woolly mammoth. I know passenger pigeons, I think, are your passion, but I'm super excited about seeing a woolly mammoth maybe one day. Where does that project lie?
So that's over in Boston. George Church leads the Mammoth Project at Harvard with a team of postdocs that are just really pushing the field forward. And they actually have in a petri dish right now elephant cells that carry about 14 to 16 mutations from mammoths, affecting traits for hair growth, fat development, hemoglobin, oxygen binding, and channel cells, and all these major traits that they're trying to influence are mainly to hit at the one question. Rather than how do we really make a woolly mammoth, it's more so how do you get an elephant to live in Siberia, where it's cold and not tropical? And that's in essence what a mammoth is. And so there are cells living right now that could become the next woolly mammoth, but his team is doing a really cool project to research them. They're going to turn those cells into stem cells and then turn them into different tissue types, skin, blood, fat, and actually see, are the mammoth mutations creating a different trait than the elephant genome makes? And so they'll compare the cell types in the different tissue forms, and so they'll be able to assess whether or not they'll have a mammoth without ever actually making a mammoth. And rather than clone the mammoth from those cells, they're actually going to be looking into interesting ways to improve upon the cloning process. And this is really cool. And this is following a project at San Diego Zoo with the northern white rhinoceros, which has only 3 living animals left on the planet.
Wow.
And they don't breed anymore, but Oliver Ryder down at the San Diego Frozen Zoo has the cells of 12 different northern white rhinoceros, and they're taking those cells, turning them into stem cells, and they've already achieved that step. And they're going to convert these stem cells into sperm and egg cells, and then create embryos through in vitro fertilization. And in theory, this should have way better efficiency. It does not require the extensive amount of egg cell donors that cloning requires. It could be just a really revolutionary way to move these things forward, especially for hard to work with animals and ethically difficult animals. And in the next, 5 to 6 years, the world should be waiting to hear about potentially a new generation of white rhinos.
Oh, very cool. Facebook, I see you have a question.
There's also a question about cloning humans, and how close do you think that technology is?
So I don't know if research on cloning humans in the United States is actually legal. And I believe it's might be illegal in most countries in the world. And I don't know if anyone's actually actively working on that research now.
Probably some mad scientist on an island somewhere.
Probably, probably, there's a mysterious island out there somewhere, or as we all know, there's probably some ship moving between international waters. From people that work with human cells, there are some attributes I know that would make cloning somewhat difficult due to the nature in which pregnancy and cell development happens in embryos. I mean, these things could be overcome clearly, but I don't think anyone in the world is really prepared yet for cloning, and we seem to do pretty well making more humans without it. So.
Now, one cool thing especially about the Mammoth Project, is that it's cool to see them and to be able to have these creatures around. But there's also a kind of ecological reason, is that the Woolly Mammoth Project wants them to graze on these grasses that are being exposed by by global warming. That they don't have really creatures up there that can do all the lawn mowing that needs to be done up there. So they're kind of and I think with the passenger pigeons as well, this isn't just like, oh, wouldn't it be nice to bring these animals back? There's kind of almost a selfish reason to want these animals back in the ecosystem, right?
Yeah, with the case of the mammoth and the passenger pigeon, they're both what we call ecosystem engineers. What they do, given their size and their abundance in their ecosystem, is actually create and stimulate the process of that ecosystem for other species. And for the mammoth, what's really awesome is just look up the work of a Russian scientist named Sergei Zimov. And he actually owns a chunk of land in Siberia, which he has named Pleistocene Park.
Okay.
And he has used grazing animals to convert tundra back into grassland, which it used to be 10,000 years ago. And he's been measuring the difference in the productivity of grassland versus tundra. And the grassland actually insulates the permafrost and keeps the soil 4 to 6 degrees Celsius cooler. And it also brings in carbon from the air, sinks it down into the soil.
Wow.
And so the tundra is one of the largest biomes on the planet. And we feel that the restoration of that grazing ecosystem, mammoths included, which help engineer that system, if we convert the whole thing, we could really change how humans affect climate on this planet in a very beneficial way.
Very cool. Facebook, I see you have a question.
Will we be reviving the Megatherium, which, for people that don't know, is an elephant-sized sloth?
Okay.
So I actually get that question a lot. And I think the key thing to remember is nothing is technically impossible, but given the current technology, it's probably not plausible now. And the main reason is the surrogate parent. So we could take living sloth cells and engineer a Megatherium genome, but the Megatherium is several thousand times the mass of a living sloth. And even actually the offspring of a Megatherium is a couple hundred times larger than a sloth. So there's no mother to give birth to the Megatherium. But as George and other scientists working on this all would say is, in 15 or 20 years, we might be 3D printing organs. So we might be able to create a Megatherium uterus in a giant test tube and raise a baby Megatherium. And at least in California, the Shasta ground sloth, which is much smaller, there's interest in restoring that animal because of the Joshua tree. So the Shasta ground sloth was a coevolutionary partner with the Joshua tree. It was the major seed disperser for that tree. And as we march forward in time, many of those trees are starting to become restricted in range, and their seed dispersal just isn't happening very well. So we had a few people contact us that have some interest in Shasta ground sloths. But once again, even that, the smallest giant sloth, is still way bigger than any living relative they have today.
Facebook, I see you have a question.
We have a viewer who asks, why aren't we curing disease with this technology?
That's actually where this technology comes from.
Yeah.
So just start Google searching this kind of thing. So CRISPR-Cas9 is actually a bacterial immune system device. When a virus enters bacteria, CRISPR-Cas9 in bacteria goes out and it chops up the virus and kills it. And people are now using this, they figured, oh, if it chops DNA, we can use this for engineering. But now people are saying, well, God, this thing is originally designed to kill viruses. Why aren't we using this to fight HIV or hepatitis C? And they've started doing that research in cell cultures. They're also using the engineering aspects of it to start working on diseases like leukemia, and other types of cancers, and creating genetic engineering approaches to help with those diseases. Now, our black-footed ferret project is actually a conservation disease project. So the black-footed ferret is an endangered species. It's still alive, but it faces a struggle with sylvatic plague, which is the same thing that gave people Black Plague in Europe. The disease came from Asia, spread over the Great Plains, and it just prevents the ferrets from living in the wild, and they don't have any natural immunity to the disease. So we're exploring ways to give them resistance to plague from their relatives, the domestic ferret, or novel ways of producing that. And we've been in meetings about that project for several years trying to design this. It's not an easy task, but we're going after it.
And then when you bring a species back, or, let's start the question this way. Is that species pass on information, from parent to child, like a culture almost, and like animals do learn from their parents or from others in their community. Now that information is kind of lost. So if you brought back passenger pigeons or woolly mammoth, would they necessarily know how to be passenger pigeons and woolly mammoth because there's no kind of a parent to teach them? Can we? Is there anything? Would they act as their ancestors did?
So with any species, there's an interplay between instinct and being nurtured.
Right.
Some behaviors that are just ingrained, such as for migratory birds. It's practically instinctive to migrate for some of them, while others actually have to be assisted by their parents to get to where they're going. And because it's different for each species, it comes down to really knowing the animal you're working with. For mammoths, the assumption is that their family lifestyle is actually very similar to living elephant lifestyles based on how their fossils have been found in family groups and how their traces over Siberia have been analyzed. For the passenger pigeon, we have the ability to recondition their parental behavior with living pigeons, rock pigeons, which are highly trainable and can be manipulated into producing tree-like breeding colonies. And the real goal here is not necessarily reproducing a duplicate of an extinct species that matches every aspect that it once was in the past. This is really about using the information that we have of an extinct species to be able to produce a viable animal that can reassume its ecological role. The IUCN, which is the International Union for the Conservation of Nature, has dubbed de-extinct animals as proxies for extinct species. It's really a form of ecological replacement, which is something that conservationists have been doing. Usually you take an animal that already looks exactly like your extinct species and behaves like them and bring them into an area. But for some animals like woolly mammoths or passenger pigeons, there is no living species that could assume that role without some facilitation. So for an elephant, an Asian elephant behaviorally and biologically is almost a mammoth. It just needs that help to be able to live in the cold. A living band-tailed pigeon eats all of the exact same types of food as a passenger pigeon, raises young as parents in a very similar way to passenger pigeons. The key difference is they don't breed colonially. They're not social. They don't come together in large, dense flocks. Now, it's likely that that behavior of social living is a combination of genetics that comes down to part instinct, but also raising them a certain way. And by raising our new passenger pigeons with rock pigeons, which do live socially, we can condition that behavior and express those genes and get the animal we want. And we'll be doing that in very large enclosures that mimic and expose these animals to a wild scene. And so a research period before we actually release what could be the wrong bird. We want to make sure that we're releasing the right bird.
All right, we're actually way over time, but I'll just ask one final question. Bigfoot, is he out there or she?
So, well, near Santa Cruz,
PC Mag exclusive coming. Yeah.
Near Santa Cruz is a town called Felton.
Okay.
And they have a Sasquatch or Bigfoot museum. A couple of guys who have been doing, going out their lives looking for Sasquatch.
Yeah.
So is he out there?
Yeah.
No, I don't think so. Probably not.
Okay, fair enough. Ben Novak, this has been an amazing conversation. Thank you so much for doing this. People at home, check out Revive and Restore. They're doing amazing work. And thank you to everyone who participated at home. Thank you to Social Pete for doing his Social Pete thing. If you like this interview, give us a like, give us a share. We really appreciate those. And check out some of our previous interviews. We do a lot of cool ones. And as always, be good to each other. Peace.